hot climate. Whereas the stem rust is much of the warmer areas, particularly central India and peninsular India are highly prone. And whereas the stripe rust is mostly in the cooler region, but once it appears, you can write the total yield loss. You can write off the crop. The important phases in Indian wheat improvement, the pre-green revolution period, importance of rust survey and surveillance, then discovery of Norrin 10 gene for semi-dwarf wheats, and also discovery of 1by1 RLS translocation from rye, and identification and cataloging of rust genes, focused gene deployment, understanding host pathogen interactions is now the recent trend, then gene pyramiding through marker-assisted breeding, and identifying neogenes and QTLs from the land races and also from the land races and the wild species, and development of the derived synthetics. The pre-green nutrition period, the tall Indian varieties were grown, and but they were low yielding, and because of the lodging it causes the damage. Then in India producing only around 10 million tons of wheat at in the year of the 1960, and India was called begging bowl to almost empty to the plenty. Now we reach the situation. But the tall Indian wheats contributed for several traits, particularly for hardiness, biotic abiotic, and also for biscuit making quality. It is the Indian wheat varieties that has contributed, and recently almost all identified QTL genes, Indian wheat varieties were the source. The breeding of rust resistance in Indian efforts, earlier the pioneer wheat breeders Howard from Pusa, they initiated the breeding program in India. They could release the Pusa varieties, the Pusa and New Pusa NP series, and later on the Dr. P.P. Paul and other groups, they contributed, and the NP 218 was one of the famous varieties. Then we use effectively the races from the Kenya, the Kenya E20, and for the black rust. But the effective way to fight the rust in India is the best way to understand the pathogen first. So when we want to be a successful breeder, first you have to understand the pathology or a pathogen. Without that, you cannot make any contribution. So this is the classical work of Dr. K.C. Mehta, the epidemiology in India. He is a famous epidemiologist. Although this theory has been well proven when it goes to sexual cycle, particularly for stem rust, but it is not applicable in India. So in Indian wheat rust mostly survives only in Nilgiris. Particularly black and brown rust spreads from the Nilgiri hills and reaches the Gangetic plain in the month of February. It has been well established. This is the rust path, or the famous, it's called Paxinia path. The spread is from Nilgiri hills, it goes up to the central India, and also it comes from the Himalayan hills, but mainly of brown and yellow, but not the black. This is the Paxinia path, and we have the strategy to develop the varieties and spread across in the Paxinia path area in a mosaic pattern, so that we can avoid any further epidemics. This is the wheat epidemiological study, and the last, particularly the turn of the century, yellow rust is emanated from Kenya in 1986, but it took almost a decade period to reach India because of the wind trajectory. It has gone up to the Iran, Iraq, and followed up to the Afghanistan, entered the Indian region, and it broke the OR9. So there was a variety called PW 343, even still it is being grown, but it is highly susceptible to the yellow rust. Then afterwards, in 1999, the stem rust race emanated from Uganda and spread to Kenya. Now it is confined to particularly the Iran region, but is expected to anytime to reach in India, not only through the wind trajectory, because there is lots of people movement across the globe. So even by human touch, it can also come. So the risk factor is very high, because almost 90% of the world germplasm are highly susceptible to the UG99 race. So that's the biggest threat. Initially it has broken the SR31 gene, stem rust gene, then subsequently 36 it evolved in Ethiopia, then when it reached Iran, Yemen, then it has broken this other gene like SR24 and so on. This is the Kenyan wheat nursery, and where we found SR24, 25, 26, 27, up to what are the genes which I am listed here, they are very effective. But the first victim was the SR24. The moment it been incorporated, it been overcome by this UG99. And after that, the BGRI, Borla Global Research Initiative, we took an initiative and make the conglomeration of the scientists working from across the globe, and we are making the stem risk baseline survey workshop. It was held in Shimla in India. Then after that, we brought the quick sets which can detect the arrival of UG99. If at all UG99 arriving in India or surviving in India, it has to survive only at Wellington, then it starts spreading to other places. So we have taken the keen initiative to effectively monitor. So around the year, we are maintaining the green crop. It's called the green bridge has been created so that we can continuously monitor. And these are the quick sets. These are number of genes, particularly LR24, SR36, and also SR31, and the highly susceptible one, the Morocco, and also the rye. We have kept as one of the checks. The important Rust pathotyping is for effective gene deployment. This is the number of pathotypes available in the blacklist, particularly now dominant one, or the 40-1 and 40A. There are two dominant races. Particularly the brown rust, 77 group, it got mutated and it has thrown out 7 variants until now. So it has overcome almost 5 genes over the period. But however, the 77-5 is a much dominant one. So the pathotype profiling, you can just see in the after 2001, much of the dominant one is 77-5 even today. So we are now taking concentration on this. It has broken this LR26 from rye, which is widely deployed across the varieties which in this country. Almost 80% of the varieties carrying this gene. So these are the dominant wheat races in individual regions. And northern India, we have 21R and also 40S119, which is virulent on YR9. But in southern India, particularly in Wellington, these are the dominant races. So over the period, you can just see the one or the other genes, a number of genes been broken periodically, overcome by new pathotypes. But however, this group, 77A group, has thrown out number of variants. So based on this, we could able to formulate a project. So we have a Pakistani Genomics program, along with the TNA is one of the partner in this program. And this is the evolution of races, 77 over the period. And this is 77, almost 75%, 77 groups, pathotypes in the field samples. Nilgiri has got the maximum variability. Then over the period, you can just see the gradual narrowing of the resistant sources. So now we are left with few genes which are very effective. So we have to take care in the future that we have to mine for the newer genes so that we can have the effective deployment. This is the situation yellow rust. Only 2 genes are effective as of now, YR5 and YR10, and of course now we have YR15 as well. Understanding host pathogen relations, that's very important to know that what type of resistant available, whether it is a slow rusting or the fast rusting group, or having the adult plant response or hypersensitive, or the type of resistance which is being classified. This is the way we classified it. And this UG19 resistant breeding at IRI regional station, Wellington, have been taken very effectively. We have made a stride and we could able to develop number of varieties. The discovery of Norinten and the 1B1R translocation from rye played a very key role. The Norinten gene, which was identified from the Japan and then were taken to USA, and the variety Bravo was developed. From there, it has been taken to Mexico. Then subsequently, in 1960s, there's released a series of semi-dwarf varieties that has revolutionized the wheat production worldwide. Then subsequently, the deployment of the gene 1B1R translocation, which has come like a cassette, SR31 for stem rust, LR26 for leaf rust, YR9 for yellow rust, and PM6 for the PM9 for the powdery mildew gene, sorry, PM8. 4 genes came along with the slow senescence of the leaf, which has contributed for the 15% yield jump in yield. That's the biggest contribution. And as of now, more than 80% of the variety cultivated in the world having the gene SR31 derived from rye. So this is the gene deployment, the dominant genes which have been incorporated in different regions, like a mosaic pattern. We are having the variety, and because of that, we could able to effectively checkmate the rust across the country. So last 15 years, almost there is no epidemics because of gene deployment. So this is a number of resistant varieties which have been classified. It's unintentional. It has come along with the semi-dwarf varieties. This is the number of genes. After that, later on, we could able to understand the genetic basis of resistance in the wheat. These are the number of genes it was available floating across the varieties which have been released earlier. But now we are having the different approaches. There are two approaches. One is the short-term goal, the near-term goal. Other one is the long-term goal. The near-term goal is for deployment of the known effective, the what you call dominant genes, because they are tend to overcome by the new pathotypes. So the risk, the concurrent risk is involved in that. However, for long-term goal, we are combining with the adult plant resistant genes. Otherwise, it's the QTL, slow rusters, so that we can put more and more gene incorporated. We are reaching to the near immunity. That's the only way effectively we can manage the rust in the future, so that no further breaking down will take place. However, we are combining one way or other along with the minor major combinations, so that we can develop resistant varieties. Then the type of resistance, there are race specific resistance, race non specific, but they are classified as what you call the vertical resistance, other horizontal resistance. I don't go much detail in that. So, but the effect is the higher level of resistance always the epicity over the lower, given longer protection, linkage drag for phenotypical markers, yield gain is possible, improvement in nutritional quality is the contribution to the major genes. However, the minor ones, they are having the near immunity and durable, no linkage drag, because none of the unwanted traits associated. For the classical example is SR31 from rye it has come, but it totally altered the quality of the atta. So that's why the modern varieties, we make chapati, it becomes very brittle. Within 2 hours it becomes brittle because of the quality drag associated with SR31. That's really a negative trait it has come. That's why millers, they don't buy SR31 varieties. That's the reason why they procure only from the central India, which is having the quality variety, which doesn't have any SR31 gene in that. The QTL response is associated with other genes, because it is like a cassette and it is a pleiotropic. It's not only one rust gene alone, it is associated, it is for the drought resistance, insect resistance, and also for the multi disease resistant varieties, multiple disease. And it is a quantifiable and also more to the gives a near immunity, no linkage drag, and CAPR. The effective genes which already we have shortlisted in the world as of now, more than 70 genes of leaf rust have been identified and cataloged in Australia, and we have the entire set with us. So out of that, these are the genes for the leaf rust which are effective in India. Whatever been listed is a red. The program already we completed and distributed the varieties across the country, almost 20-40 centers. So they've been provided with the variety, and the one which is given the pink, they are being currently incorporated, and you can just see almost 99% of the genes all from the alien sources. That's called secondary and tertiary gene pool, not from the wheat. In wheat, only one gene of leaf rust is from LR34 or something minor genes. None of the major genes has come, but the SR2 is one from the stem rust, remaining all from the alien sources. This is the number of stem rust genes and yellow rust genes we are already maintaining there, and these are the genes identified as resistant to E99 that we have taken into the program. But what we have done is, we have taken nearly more than 30 varieties which have been carefully selected across the zone, nearly from the 6 zones, each almost 5 varieties have been taken for incorporation of the genes, and that's been effectively done at Wellington in a very planned manner. But the initial concept was to have the single gene concept, but over the period that has been overcome by new pathotypes, so that was the risk involved. So LR9 before deployment, it has already been overcome by new pathotype 77-7. But LR19, I think it is going to be a replacement for SR31. This is also linked to the slow senescence, increased yield, increased number of tillers per plant, and also spikelets per spike. That's the trait associated. So now we are exploiting it very effectively now. And over the period we lost the track because the material supplied by Australian, that McIntosh, it was not carrying that 19. It was proven only after 5-6 years of incorporation. Later on we shifted to the newer sources. The LR24, it is a very classical gene which is giving the resistance across the country more than 30 years. but elsewhere in the world like Australia and also US and Mexico have been overcome by new pathotype, but not in India. However, the linked gene SR24 have been overcome by 40-1 pathotype at Wellington in the year 1984. And other gene, this is the one, the classical gene which I told, that's SR13 from rye. It's very effectively utilized almost across the globe. That's the risk which we have taken. UG99 is overcome. Now the entire world production is under threat. So everybody is rushing up to diversify the genetic base of resistance elsewhere. And came the very effective gene LR28 from Agellos peltoides from again goat grass, or it's called it's Agellos group. It's a very effective gene, and we are effectively making it. 32, it is also from Scorosa, and that is also a slow rusting type, but however a very effective gene, combines very well with other genes. Then LR37 has got the advantage linked to 3 genes. The one is stem rust, another is yellow rust, and it is temperature sensitive. And yellow rust gene not effective under certain varieties because of the gene action. Then gene pyramiding and conventional method, both we are using it for developing varieties. These are a number of big varieties which we have released from Wellington for almost covering all zones. The last year we have released a dicoccum for entire country, and also HD 2967 to Punjab is fast picking up. This is the number 1 variety now cultivated in Punjab. And for the UG99 specific, we are combining both the slow resting and also the major gene, particularly the slow resting SR2, very easy to interrogate because it is linked to a phenotypical marker called pseudo black chaff. And I think this has been well done by one of the students from TNAU, Philomen Juliana. She was in USA along with me, Cornell. They were a very good job. Then the gene pyramiding, these are the number of genes which are taken, and for UG99 specific, we are already ready with the varieties. For each zone, we have a variety already given and distributed to the people for utilizing in the breeding program, or else we can release it as a commercial cultivar once it has been put in the trials. Then the serial derived another gene LR45 is a very effective, good combiner, and it's also associated with the phenotypical marker. You can just see very well wherever that gene has been incorporated, it is having the pink horns. It appears for the only 1 or 2 weeks during the duff stage and it disappears. So it's a very effective phenotypical marker. So it's a very cost effective incorporation. The Agellos peltoid derived gene, the linked gene, this also we are using, but we have withdrawn this gene. Later on we found that it is having the yield penalty. So that's the reason why we are not using it. However, to checkmate the rust in the rust path, we are developing varieties in a mosaic pattern and incorporating and release the variety. Then we already released the dicoccum jointly with the TNAU as Code W2 for Tamil Nadu and the non-traditional areas. And recently one, as I said, the coupling, what you call Nilgiri coupling, it is SW 1098, and this is fast picking up now. Then another one is 5207. This year it's been identified by TNAU jointly. Then we are going to release it as a Code W3. This is a joint contribution by TNAU and the thing. But the very interesting thing is it is having a linked gene for the 3 traits, for YR15, LR24, and also SR2. Then 5216 for Southern Hills we released, and we are maintaining more than 3,000 assessments of wild species and wild related primary, secondary, and tertiary gene pool. And I should be very thankful to Dr. A.J. Prabhakaran. When he was with my colleague, he imported from USDA, and we are carefully maintaining the largest collections. So these are the linked gene varieties which already were released commercially for cultivation. This is a very famous variety and the number 2 variety in Peninsular India, which has been carrying two sets of genes like LR24, SR24, the SR31 complex. Then we are effectively making the marker selection, and the newer gene combinations which are taking up now, these are the gene lists, and this the exploitation of linked genes that will give you the what you call multi disease resistance. Then we are identifying novel traits from the wild species for high tillering, particularly from morography and also from Agellos groups. And this is a number of genetic stocks in the recent times we registered with the NBPGR, so it's been effectively utilized by the breeders. And we are collaborating with Australia in identifying newer genes, particularly from Indian land races, and we are almost closing in on the 2B, 4B, 5B, and 5A. So we are going to come out with the second APR gene in the world, which is shortly going to be published. And the durable resistance program, we are part of it in the programs, but the frequency you can just see. This is a single gene, 2 gene, and 3 gene combinations already been deployed in this country, and This is the frequency of cultivars having leaf resistant genes, and this is the frequency cultivar carrying LR 13 and 23 and 26. And this is the what we analyzed last year. We come out with the review paper in plant breeding and genetics. That's the LR24 deployment across the country. This many number of varieties carrying this SR24 and is contributing much. So this is my conclusion, that over the period, particularly last 1 decade alone, 40% yield gain have been made because of gene deployment. That's because of the release of cultivation of the high yield and disease resistant varieties, efficient vibrant coordination, and also the gene identification. The future lies with much of the wild species and the wild species. Now we are exploring it and we are maintaining carefully and we are making it. The other one is a synthetics, a derived synthetics, the cross between durum and it's from dicoccum. Dicoccum and durum based from agelofs is being utilized. So my conclusion is the survey settlement mechanisms should be very good. The pyramiding of major genes and minor genes and use of conventional and also molecular breeding to effectively release the varieties in hotspots and hotspots exploitations, and then fine tuning of SRT and developing varieties of known disease resistance and the exploitation of positive traits associated with the pyramiding of genes. With this, I thank the organizer for giving an opportunity. Thank you very much, sir.
Thank you, Dr. Sivasamy, for your masterly presentation, and it is open to the question. For the last 3 or 4 years, I have had a wonderful opportunity to utilize the facilities of Dr. Sivasamy in his research field, and these are all the fields he chose. So, a very large field board with ever so many genes. One day, not out of distrust, but out of curiosity, I asked bluntly Dr. Sivasamy, will you be able to remember all the genes? Yes, why not? And he was telling all the genes in an offhanded manner. That is the thoroughness of his acquaintance. And just like a rust spread from our Nilgiris and from our hills to entire country, his knowledge spreads for rust resistance grows. Thank you.
Thanks for your
open for discussion.
Thank you.
Dr. Sivasamy, will you give a little more light on the situation of UG99 and in the Indian scenario, because it is a threat to India. Yeah. How preparedness we have made to face it?
Yeah, the thing is, we have developed a number of stocks, but the problem is the replacement ratio. Seed replacement is the biggest thing. To almost to supply for 29 to 30 million hectares. So seed increase, it will be a big problem. Otherwise, we have the stock. India has taken a pioneering step, and we are much ahead than even USA. So that is the reason why I have been called to Cornell to contribute for them, and we have already given the spring week background to the winter wheat. I completed my work there, and India's, I should say, we are comfortable one way. The main reason is India is the only country in the world which is cultivating 3 species of wheat: durum, dicoccum, east ibum, in a mosaic pattern. Particularly in the Puccinia path, we have the varieties of dicoccum durum, which can probably give a buffer before it is, because in any case it has to come to Wellington, and from there survive it goes to the central India and the Ganges plain. But there's already a contradiction between the scientists. The one set of scientists like Dr. Nagarajan is put forth that UG19 will never reach India. Same way we were complacent earlier for yellow rust, but we paid the heavy price. Almost a decade ago, nearly about 10 million tons food grain we imported, which cost the exchequer to the tune of 10,000 crores. So we cannot remain complacent. That's the reason why we are giving much emphasis, although many people are criticizing it. But we are diversifying. We have the gene sources. We have the stocks already developed. So I hope India can manage it.
This topic will be understanding on the host plant resistance and its exploitation in crop improvement.
My colleagues and my student friends. So we had 2 nice talks about the utility of molecular markers, one from Dr. Aswath and another from Dr. Sivasamy, how these molecular markers can be exploited for the crop improvement. And this story will be something different from their stories. So now here also we are going to see some of the aspects related to utility of molecular markers with reference to resistance to insect pests in plants. Yes, Sivasamy was telling about the easy usage of molecular markers in deploying the genes for resistance to rust. We have problems with insect pests. So the problem of post-plant resistance with diseases is much more easier than with insect pests. So that is the thing I am going to talk, and how these markers can be used and what is the importance of biology we have to deal. That is the main thing. So here you are having a set of insect pests and their feeding behavior is entirely different. So when the pests are attacking the plant, there are different modes of defense. One is constitutive defense and other is induced defense. And when these defenses can influence the insects, herbivore settling, feeding, oviposition, growth and development, fecundity and fertility. All defenses, whether constantly or transiently expressed, are costly to the plant because it is something, a great loss to the plant when they are trying to defend the insect pest. Next. So these are all some of the plant defenses which the plant have for the insects when they are attacking. Next step, please. But when these insects are attacking on the plant must attain a balance to ensure survival from immediate and subsequent attacks without sacrificing the plant vitality, longevity and reproduction. And it has to deal with the supply the plant with flexibility to achieve these goals. If this is not possible, the plant cannot survive. So these are all some of the components of defenses which were explored in plants for various pests. So why I am trying to tell all these things, we will see one by one. Next slide, please. The importance of plant resistance is when we are dealing with the insect pest and controlling to have a control, we are following different methods, and host plant resistance is one of the best methods available for controlling the insect pest, because here we are having lot of problems and we have to have the control over the environment when we are doing this. And this is an inherent property in the plants which can be exploited when we are dealing with this host plant resistance. So this study on plant resistance has been done in many fronts, and the coordination among these fronts is sometimes impressive, but at other times seems non-existent. So for example, when Sivasamy was telling about the rust resistance, there was a clear-cut coordination, but when we are dealing with the insect pest, this is not happening. So if you are getting into the example what I am trying to explain, then you will be understanding the problems. And most of the times, we are doing our research just to confirm the results already published. We do the confirmatory research rather than going for exploratory research. So variable plants and herbivores in natural and managed system, because we are doing all these things under the agricultural system. But when we are seeing at the natural system, the resistance becomes very complex and uncontrollable. So what is the relevance of plant resistance? This indicates what are all the reasons we have to try on the plant resistance. And this study, the exploiting the plant resistance for the crop improvement, has been started in 1955 by Painter, and these were the crops dealt by him while he was exploring the post-plant resistance in different crops. And he established 3 components: antisynesis, antibiosis and tolerance. This everybody is told from our undergraduate class. But if you see the original paper of Painter, the resistance, these things were given as a components, but over a period of time, our entomologists changed these components as mechanisms of resistance. So this is the thing happened. Actually, Painter told they are components of resistance. And in later period in our classes, we were told about that this is mechanism of resistance. Next slide, please. So there are as many definitions of resistance as there are scientists studying the phenomenon. So if you see the host plant resistance definitions, there are empty number of definitions available. The definition of resistance is context dependent. Genes conditioning the presence of chemical or physical attributes that interfere with the ability of the herbivore to utilize the plant. Resistance to one pest may be due to the presence of the attribute, and for the another may be absence of the attribute. Example, for example, cucurbitaceans, presence of cucurbitaceans make the host susceptible to cucumber beetles, but confers resistance to mites. So these are all the situations, but when we are studying, we will be in a complex situation, we may not be knowing what is the actual reality. Next step. So these were the refined definitions of antigenesis, antibiosis and tolerance by Smith and 1989, and now he has a very good book on host plant resistance in 2005, and which is freely downloadable from the Google books. Next slide. So this is a diagrammatic